Hello there. Today I will be explaining about Elisa. In the slides to come, I will be talking about Elisa, the procedures, the materials required, the types of Elisa, the advantages, and also the applications as well. The abbreviation, that is the name of the procedure that I'm explaining. It is called enzyme link immunosorbent assay because the secondary antibody is linked with an enzyme called with an enzyme and it is used in the ELISA procedure. What is ELISA? First of all, before we talk about ELISA, we need to know what antibody and antigen is. An antigen is a toxin or foreign substance or material that can infect our body, like the bacteria or the toxins that are being produced by the bacteria. The antibody is produced as a result of the antigen which infects our body. The antibody and the antigen forms an antigen-antibody complex. So the ELISA procedure is used to determine or to quantify the amounts of antigens that can be found in a specific sample. But it doesn't just stop to determining and quantifying the amount of antigen. It can also be hormones, antibodies, and many other materials as well. The basic procedures of ELISA. This is the ELISA plate. It contains 96 wells, and in each of the wells of the wells are being bounded by antigens. And when the sample is added, there is a formation of antigen and antibody complex. The wash buffer is used to wash off the antibodies that are not being bounded to the antigens. And later, the secondary antibody is linked by an enzyme. That secondary antibody is added to the sample, and when it is added, the wash buffer is also added in order to wash and flush off the secondary antibodies that are not bounded to the primary antibodies to form also a complex. And finally, the substrate which reacts with the enzyme is the enzyme on the secondary antibody in order to produce the final color of the reaction. Other than antigens, antibodies can also be bound to the walls of the ELISA plates, like in sandwich ELISA. In sandwich ELISA, the ELISA plate, the ELISA wells are being bounded by antibodies. The antigen then bound to the antibody to form an antigen-antibody complex. The secondary antibody, as I mentioned earlier, which contains the enzyme, which is bounded by the enzyme, is then attached to the antigen here. And the substrate is later added, that is the chromophore. The substrate reacts with the enzyme in order to produce the color of the final procedure. The materials needed in an ELISA procedure. These are the antibodies, antibody 1 and antibody 2. Antibody 2, as I mentioned, it is bounded by an enzyme. That is why it is called, the procedure is called of this ELISA, it is called enzyme linked antibody, enzyme link immunosorbent assay, and an antigen which is produced as a result of foreign materials or bacteria and many other microorganisms infecting our body. The polystyrene microtiter plate It is the ELISA plate that we use to process or that the reaction is being carried out. The blocking buffer is usually the bovine serum. The bovine serum is used in order to have, in order to prevent the formation of a false negative results. The bovine serum, after the antibody is added to the is bounded to the walls of the ELISA wells, the bovine serum is used because in between the antibodies there are spaces, so the bovine serum is used to fill those spaces in order for the secondary antibody, which contains the enzyme, not to be bound to the walls of the ELISA well, for the form which can lead to the formation of a false negative result. The washing buffer is also used to flush off the antibodies, which can be the primary antibody or the secondary antibody, that are not bounded to an antigen. The substrate and the enzyme react in order to form the color formation. These are some of the enzymes that are used in ELISA procedures, such as horseradish peroxidase, alkaline phosphatase, beta-galactosidase, lactoperoxidase, and tetra-methylbenzidine. But usually in many ELISA procedures, the horseradish peroxidase is used. Why is this enzyme specifically used in most ELISA procedures? That is because it has the ability to amplify weak signals. Due to this, it can detect small molecules that are found in the reactions, such as the antigens. The types of ELISA. There are basically four types of ELISA: the direct and indirect ELISA, the sandwich ELISA, and the competitive ELISA. In the indirect ELISA, the wells of the microtiter ELISA plate is bounded by antigens, and the secondary antibody, which has an enzyme bound to it, it forms a complex with the antigen on the walls of the ELISA plate. The substrate, after adding the substrate to the wells of the ELISA plate, it will form a product which leads to the formation of the color change, depending on the quantity of the antigen in the plate or in the reaction. Indirect ELISA. In an indirect ELISA, just like the direct ELISA, but it has some little bit of differences. In an indirect ELISA, the antigen is bounded, forms a complex with the primary antibody, such like this one, and the primary and antigen complex, the complex formed by both these ones, later bound to a secondary antibody. The secondary antibody has an enzyme bound to it. This enzyme reacts with a substrate. As you know, enzyme reacts with substrate is like a key in a lock. They fit together and a reaction which leads to the formation of products, as you can see here. Sandwich ELISA. In sandwich ELISA, as the name implies, it is like two antibodies having in between them an antigen. This is the antibody or the primary receiver. The antibody or the primary receiver will form a complex after mixing it with the antigen to form the antibody-antigen complex. The antigen and antibody complex will then, the secondary antibody will then be bound to this complex, and after adding substrate to this complex, that will lead to the formation of the products, that the color is going to change. And finally, the competitive ELISA. During a competitive ELISA, antigen and an antibody are incubated in to, at a degree of 37 degree. In the walls of the ELISA plate also, the same antigen is being used. Why do we incubate the antigen and the antibody in at 37 degree? This is because we want to create an antigen-antibody complex, and this complex will then be added to the ELISA wells, and when it is added, because of the high affinity of the antigen-antibody complex and the antigens on the tips, so this complex wouldn't be able to attach or to form another complex with the antigens found on the walls of the ELISA plate. As a result, after it being washed off, it wouldn't be in the reaction anymore. So the antibodies in the reaction that were not able to bind to the antigen will then bind to the antigen in the walls of the ELISA plate. So this means the higher the antigen in the sample the lower the the lower the amount of antibody that are going to be bound to the walls to the antigens in the ELISA plate. And then the secondary antibody, which contains the enzyme, will be bound to the primary antibody. And when it is washed off to remove the excess secondary antibody that were not able to bind to the complex, if the substrate is added, then there will be a color change. The color change depends on the amount of antigen in the reaction. Some importance of the ELISA. It is quick and convenient compared to some biochemical reactions. It detects low concentration of antigen because of the enzyme that is used. As I mentioned before, it has the ability to detect small molecules in a reaction and it amplifies them. It is safe. Why? Because it doesn't contain any radioactive substances. The radioactive substances, some of them usually are hazardous to our health. It is used in variety of tests because it has a high sensitivity. It has the ability to detect even small molecules, small biochemical molecules, and it has strong affinity because of the antigen-antibody complex forming together. Some of the disadvantages of ELISA: it can form a fake positive result. What is a fake positive result? A fake positive result is a result which is negative and it is termed as a positive result. This is because if the blocking solution, as I mentioned before, the bovine serum that is added to the sample or the reaction in order to block the empty spaces found in between the antibodies, therefore the secondary antibodies will be bound to the walls of the ELISA, and if the substrate is added, it will form a fake positive result. An enzyme substrate when an enzyme and substrate are added together, the reactions immediately start. So therefore, after the plate should be run immediately in order to avoid that, or in order to avoid having negative, fake positive result. Some of the applications of ELISA in today's world, it is used in food industries and some in medical and laboratories, in hospitals as well. In terms of health or hospital, it can be used to detect AIDS, HIV, and AIDS. It can be used in blood testing and also pregnancy test as a home-based. And in food industry, it can be used to detect food allergies. Thank you for listening.